Okay, I want you to think back to yesterday. Glycolysis, breakdown of sugar, splitting of sugar. Where in the cell does it take place? Sam? It takes place in the cytoplasm. Because it takes place in the cytoplasm, is glycolysis an aerobic process or an anaerobic process?
Anaerobic.
It's anaerobic. What does anaerobic mean?
Without oxygen.
Without oxygen. Why do you, as a mammal that's living on an Earth that's 4.5 billion years old, has plenty of oxygen in the atmosphere now, why do you, right now, as I speak, perform glycolysis, which is anaerobic? Anybody help her out? Give her some support?
Our ancestors didn't have oxygen, so they had to create energy without it.
When you say our ancestors, you mean grandma?
No.
What do you mean?
Like the cells and the organisms that were here, like, they didn't have oxygen.
The original life forms on Earth, the original prokaryotes, the single-celled organisms, bacteria and archaea that inhabited the Earth, they're the only things here. They existed when there was no oxygen in Earth's atmosphere. In order for them to survive, they need energy. And therefore, an evolutionary, not an evolutionary, but a process to make energy had to exist in the absence of oxygen. And that was glycolysis. So those first life forms used glycolysis. It was an anaerobic process. The fact that we have glycolysis today is evidence that all life on Earth evolved from those first life forms that were around 3.5 billion years ago, roughly. Okay? So I'm going to write all that down. We'll do a quick review, and then we're going to get into the new material for today, and we're going to map out the entire goings-on of glycolysis. All right? So let's do it. Hold on. Oh, 1 second, guys. I don't know why it's not putting the line paper on. There we go. Okay. So this is going to be cellular respiration, part 2, glycolysis. I will be writing bigger than that, so if it looks a little small to you, that's just because I typed it. I will be writing bigger. All right. So as we just said, glycolysis. If you were to mispronounce it, you might say glycolysis, but if you say it like that, you're actually separating the prefix from the suffix. The prefix glyco means sugar. The affix, or excuse me, the suffix lysis means splitting. Glycolysis is the catabolism, the breakdown of sugar. As you recall from yesterday, the way that sugar gets into your cells, you first have to eat it. You then digest it down to its smallest, simplest form. So who could tell me, why do we have to digest any type of sugar down to its monomers? Why can't the disaccharide work? Why does it have to be the monomers, the monosaccharides? Anything that is bigger than the monomer is too big to diffuse through your intestinal lining and into your bloodstream. You have to get it down to its smallest form. And when I say smallest form, I don't mean the elements. I don't mean carbon, hydrogen. I mean its smallest molecular form. And that would be the monosaccharides if they're carbs, the amino acids if they're proteins, the glycerol and fatty acids if they're lipids, even the nucleotides if they're nucleic acids. Once it's in your bloodstream, you're hyperglycemic. What hormone lowers your blood sugar when it's too high? Insulin. And then insulin escorts your sugar that's in your blood into your cells. Okay? And now the sugar is in the cells. Remember that glycolysis is an anaerobic process. It occurs in the cytoplasm of cells. when we actually get to the real cell respiration, it starts with this. Now, if you want to get technical, this is not part of cell respiration. This is like a prelude to cell respiration, because the word respiration means oxygen. You're not going to hear oxygen at all today, except for right now. You have something to say? All right. So what I would like to do first is I want to go over a few words that you guys are going to need to know for today. So words to know, there's 3 of them. You may have heard of them. You may not have heard of them. The first word is going to be phosphorylation. It simply means phosphate is added. This marker I'm holding in my hand, this is a phosphate group. Sam, you ready? Catch. Catch. Good job. She just got the phosphate. We would say that she was phosphorylated. I phosphorylated her. Here, throw it back. She just phosphorylated me back. This is a phosphate. So whenever a molecule receives a phosphate group, it has been phosphorylated. You're going to hear that word today. Now, for those of you that have taken chem, these should be 2 words you have heard of before. Oxidation. Some of you might have just gone, ugh. Oxidation simply means loss of electrons. And what's the opposite of oxidation? Losing electrons is oxidation. What do you call the gaining of electrons? Start. Reduction. Now, for my students in here that have actually taken chemistry before and passed chemistry, can you explain to those who have not, why do you call something reduction if it's a gain of electrons? Anybody tell me that? No? What is the charge of an electron? It's negative. So if you were to receive 2 electrons, 3 electrons, 4, whatever, would your charge go up or down? That's why it's called reduction. Because although you are receiving electrons, your overall charge is decreasing, and it's becoming more negative. That's why it's called reduction. Okay, any questions there? All right. So, words to know. Now, next one here is going to be molecules to know for today. Some of these you have heard of, some of these you have not. Number 1, glucose. What is the chemical formula for glucose? You should have this memorized. Hannah? C6H12O6. It is simple sugar. It is a blood sugar. It's a monosaccharide. Now, you will find out today that glucose is going to be broken down into an even smaller sugar called pyruvate. Pyruvate is C3H4O2. You will find out today that glucose is metabolized, or I guess the proper word is catabolized, into pyruvate through the process of glycolysis. Okay? Number 3, ADP. What does ADP stand for? Jasmine? Adenosine diphosphate. Adenosine diphosphate is like a dead battery. What do you have to do to ADP it in order to get it to become energized? What is it missing? It's missing a 3rd phosphate, and that gets us to number 4, ATP. Adenosine triphosphate. Now this is energized. It is ready to pass on energy to any cellular process. As you learned in Unit 2, you drop off that 3rd phosphate called the terminal phosphate, and it's going to be able to energize any sort of active process. All right, now we got 2 more, but it's really kind of like ADP and ATP. If you guys pay attention to this, what do you get when you have ADP plus a phosphate? What do you make? You make ATP, of course. Well, the next one is going to be, I have a bad feeling since first period that a lot of you are not going to understand this. I don't know why. I hope I'm wrong. I just have a feeling. And I don't want you to play into that. I want you to prove me wrong. Throw it right back in my face. Say, no, it's easy. We get it. This last one is going to be called NAD+. I introduced this to you last week. I think it was last Thursday. You do not need to know what the NAD+ stands for. If you really wanted to know, it's called nictitadenine diphosphate. That's what it is. Blech, it's a mouthful. You don't need to know what that stands for. Here's what you need to know about it. This is an electron carrier. It has the potential to carry 2 electrons, 1 hydrogen ion. I'm going to demonstrate this to you like as a little pantomime up here in class. But before I do, I want to do a little chemical equation with you all. When you have NAD+, you ready? And you add a hydrogen ion, what would that become? Don't overthink it. Go with your gut. NAD+, plus H+. NADH, you're right, keep going. NADH, what's the charge? You've got a positive and a positive. 2 positive. Have I lost anybody yet? Does that make sense? Okay, now, what would happen if you added an electron to that? What would it become? What would happen to the charge? Your NADH2 plus an electron. Jasmine? NADH plus. The charge went down 1. Okay? Now we're going to add 1 more electron. What will happen to the molecule now? NADH plus an electron. Hannah? That's it. So, guys, there's one chemical equation here. Here's your second one here. And here's your third one here. These are known as coupled reactions, where the products of reaction A is a product, the product of reaction A is a reactant to product B. Let me rephrase that. The product to reaction A is a reactant to reaction B, and the product of reaction B is a reactant to reaction C. This is known as coupled reactions. And the end goal is that, NADH. This is an electron carrier. So I'm going to switch over to the camera now, and just for the kids at home. And I'll put this back up. I want them to be able to see this. Okay, let's see if I can do this. All right, so got the camera on. Try to make it big here. Okay. All right, you ready? You got these two. You two are going to be my guinea pigs. Ready? I need your help. All you have to do is sit there. All right, guys, I want you all to play along here. These are hydrogens. What are these? Right. These are electrons. What are these?
Electron.
Okay. Now, I am currently NAD+. I am going to pick up 2 electrons and 1 hydrogen. What am I now? But I have these 2 electrons. I'm just NADH. If I was NADH2+, I would just have this. But I also have 2 electrons. You remember that?
Yeah.
All right. So I have 1 hydrogen. I also have 2 electrons. Now, I took electrons from her. Did she get oxidized or reduced? She got oxidized. What did I get?
Reduced.
Right. So when you make NADH from NAD+, this is a reduction reaction. And when you go from NADH back to NAD+, this is oxidation. You're going to see it's going to be going back and forth. So I take a hydrogen and I take 2 electrons from here and I bring them over here. I drop them off. Now I dropped off those 2 electrons. What happened to her? She received those 2 electrons. She got reduced. What happened to me? And so what molecule am I now? I'm NAD+ again. So I'm going to go right back over here and do it all over again. 1 hydrogen, 2 electrons. What am I now? NADH. What happened to me? I received electrons. I got reduced. What happened to her? She got oxidized. I'm going to bring these over here. What happened to her? What happened to me? What molecule am I now? NAD+. Let's do one more time. I get my hydrogen. I get my electrons. What molecule am I now? NADH. What happened to her? Oxidized. What happened to me? Reduced. I bring them over here. I drop them off. What happened to her? What happened to me? What molecule am I now? This just happens. It just goes in a circle. It's recycling. Now, you might be wondering what's happening to her. This is what we're going to learn next week, like Monday or Tuesday. It's called the electron transport chain. It's the 4th step of cell respiration. You're collecting a bunch of electrons for like the grand finale of cell respiration to make a bunch of ATP. That's what it's mainly used for. So whenever NADH is carrying 1 hydrogen and 2 electrons, it's going to take them to the electron transport chain, and it's going to just let the electron transport chain keep collecting them and keep collecting them until you have a lot to make a ton of ATP. Okay, let's keep going. So let's go ahead and map all of this out. This is where you're going to need a whole sheet of paper. Start at the top. You're going to work your way to the bottom. Don't write too big. You're going to run out of space before you know it. So this is going to be the process of glycolysis. You guys not plugged in yet? Those are called coupled reactions, where the products of the first reaction end up being the reactants of the next one. It's kind of like in school. Your education from 9th grade, when you're all done with 9th grade, all the new learning that you have, all the new smarts and intelligence and all those facts you learned from 9th grade, that's your product of 9th grade. Go over the next summer, That turns out to be the reactants that you need for 10th grade. Then you learn a whole bunch of more stuff in 10th grade. At the end of your 10th grade year, that's your product, all the new stuff you learned in 10th grade. But when you go to 11th grade, your 10th grade products become your 11th grade reactants. It just keeps going. Those are called coupled reactions. All right, guys, here we go. Now, the process of glycolysis, the way this is going to work is you're going to see me do a lot of these. These little red circles, this represents a carbon atom. Okay? I'm not going to draw hydrogens. I am not going to draw oxygens. I'm just going to focus on carbon. So, let's begin. This is glucose, C6H12O6. This has just entered the cell. This is ready to be metabolized to produce energy. Okay, the first thing that happens, 2 ATP are used to get started. There we go. These ATP molecules are going to phosphorylate the glucose. Duncan, what does it mean when I say the ATP molecules are going to phosphorylate the glucose? They're going to add a phosphate on each side. You have the ATP on the right and the ATP on the left. So let's see what that looks like. So now we have a glucose molecule with 2 phosphates. It has been phosphorylated. This is called phosphorylation. Okay. Next, this molecule is now going to split. But you still have 1 phosphate on each end. This molecule is called G3P. If you really want to know the name of it, it is glyceraldehyde 3-phosphate. You don't have to know that name. That's something that you would learn in organic chemistry in college. And once you're done with that, you've pretty much completed the first half of glycolysis. And this first half has a name. It is called the energy investment step. When you invest into a stock or into a company, you have to spend money, but you're hoping to get more money in return. If Sam invests into an Amazon stock today, she puts like $2,000 into Amazon today, and she doesn't touch it for 20 years, that $2,000 might become $35,000. That's a heck of a gain. That's an investment that worked out for you. Well, in this step, you're investing energy. How many molecules of ATP were used here as an investment for future energy? 2. So you basically just spent 2 ATP. So as of right now, you are at a -2 ATP deficit. You're at a loss. You're in the red, as they say. You're spending more than you're making. We're going to fix that, though. We're going to fix it. So this is called the energy investment step. Now we're going to move into the energy harvesting step. Okay? So let's do it. The first thing that happens, guys, is NAD plus comes into the picture. How many electrons can NAD plus carry? How many hydrogen ions can NAD plus carry? 1. So it's going to come in here, and NAD plus is going to oxidize G3P. When I say oxidize, does that mean that NAD plus took electrons from G3P, or NAD plus added electrons to G3P? When I say that G3P got oxidized, does that mean that NAD plus took electrons from G3P, or NAD plus gave electrons to G3P? It took them. Okay. Now also, an inorganic phosphate is going to join the catabolic pathway. I want you to take a look at G3P. How many phosphates does each G3P have right now? 1. But as you can see here, there's another phosphate coming in. So how many phosphates are going to be bonded to them now? 2. This molecule is called BPG. If you want to know what BPG stands for, it's an organic chemistry name. It's called 1,3-biphosphoglycerate. You do not need to know that name. BPG will do just fine. So to recap, here's what we've done so far. All of that. Okay. Now, we need what do you think those phosphates are going to be used for? Anybody want to take a guess? What do you think those phosphates will be used to make? Remember, the whole point of this is to make energy. ATP. Yep. ATP. So let's do that. We have ADP, ADP. It took each ADP takes one of those phosphates, and you now have 3PG. But this process, this is really important. This is called substrate level ATP synthesis. To synthesize means to create. There's two ways to make ATP. One of them is to make ATP directly from the metabolism of a substrate. In this case, the sugars, the carbon molecules. That is called substrate level substrate level ATP synthesis. The other one is made in the electron transport chain. You're using pumps. You're using electron gradients to make ATP. We will get to that later. But when you are making ATP directly from the metabolism of organic molecules, that is called substrate level ATP synthesis. There's another type of ATP synthesis that we will get to later. And so now you do not have BPG anymore because it's missing a phosphate. This molecule is called 3PG. I had a student in first period, I had a student in second period, I had a student in fourth period ask this question. Some of you might be thinking this. What is the difference between G3P and 3PG? There's been at least one student in each period that's asked that. Take a look at your image for G3P. Now take a look at what you have for 3PG. What's the difference? They look the same. Well, what happened to them between G3P and 3PG? Well, if you look at NAD+. NAD+ oxidized it. It took away a hydrogen, it took away 2 electrons. So it's not the same thing anymore. It does have the same amount of carbons, but it's not the same entire molecule. Okay, now, we're almost done. After you form G3P, a water molecule is going to break off of each one, 2 hydrogens and 1 oxygen. So in this step, oxidation occurs by the removal of water. Now you don't have 3PG anymore. You just lost a hydrogen and you lost 2 hydrogens, you lost an oxygen. So now this molecule is called PEP. I want you to notice that PEP has 1 phosphate. Let's see if you guys are keeping up here. This is 3PG, 3 carbons, 1 phosphate. This is PEP, PEP, 3 carbons, 1 phosphate. Why are they different? Dark? I'm sorry? It just shows the carbon. What changed between 3PG and PEP? Anna? You lost 2 hydrogens, you lost 1 oxygen. That might not seem like a lot, we didn't even draw that, but they're still gone. You lost 2 hydrogens, you lost 1 oxygen. It's not the same thing anymore. Just because the carbon numbers stay the same doesn't mean it's the same. Okay. We have 1 more phosphorus or phosphate on each PEP. We're going to take those away with ADP. Okay, this last molecule, this is the finish line for glycolysis. I want to include that this is substrate level ATP synthesis. And these last 2 carbon molecules are called pyruvate. The chemical formula for pyruvate is C3H4O2. C3H4O2. H4O2. Okay. What was the name of the first half of glycolysis? Energy investment step. This is called the energy harvesting step. This is like where you cash in your stocks. So Samantha, she put in $2,000 of stock when she was 16. She takes it out when she's 35 years old. It's $40,000 now. She's going to cash in. She's going to take all that money and run. Energy harvesting step. How many total ATP were made in this step? Not the whole glycolysis, just this part. Take a look at your drawings. How many total ATP? No. Look again. You got 2 here, here, and here. And you've got 2 here, here, and here. That's a total of 4. So our total net for this part is positive 4 ATP. So let's do all of glycolysis. All right? In part 1, you made negative 2 ATP. In part 2, you made positive 4 ATP. What is negative 2 plus positive 4? Your total gain of ATP in glycolysis is positive 2 ATP. This is your net total so far. And it's also worth saying that you made 2 NADHs. Those are going to go to the electron transport chain, which we will not get to for several days. Okay. Now, see a lot of pens moving. I'll wait for you to catch up. I want to bring up a point that often comes up in every period, but no one here is asking. Maybe you're thinking it. Here's what I sometimes get every year. You ready? Mr. Thorson, how come if glucose is C6H12O6 and you split it into pyruvate, why aren't the pyruvates C3H6O3? So I want to ask you, anybody in here, why is pyruvate not C or C3H6O3? Start. You forgot the water. That's wrong. When you take out water, water is removed from each of them. You get C3H4O2. C3H4O2. That's why. Because if it was C3H6O2, just imagine that, C3H6O2, you take away 2 hydrogens, it goes from H6 to H4. And if it's C3H6O3, there's 1 oxygen in water. O3 minus 1 is O2. So C3H4O2. That's why. All right, so let's put a cap on this, guys. Let's talk about your inputs. I want to hear you. How many glucose molecules went into the reaction? 1 glucose. How many ATPs went into the reaction? How many ADPs went into the reaction? 4. That 4P means phosphate, inorganic phosphate. How many NAD pluses went into the reaction? 2. All right, now let's do output, okay? How many pyruvate molecules came out? How many ADP molecules came out? How many ATP molecules came out? Four ATP. And how many NADHs came out? And not that it really matters, but what the heck? How many water molecules were made? 2. I'm going to go back up there. I'm going to help you guys count your score. Once you're done writing this down, let's go back to the very top. I'll use different colors for different molecules. Okay? So, let's color coordinate. We all like to color coordinate. We are going to make glucose yellow. We'll make ATP green. We'll make ADP blue. We'll make NAD plus orange. We'll make NADH silver. We'll make pyruvate brown. And water really doesn't matter, so I don't care about it. Okay, let's go to the very top and keep score. Wait for you all to finish. We have about 5 minutes. I want to make sure you guys can see why we had the right numbers. That's all. We're just going to double check our numbers. That's all we're doing now. Okay, let's go to the top. I only see glucose in one spot, right here. There's our glucose. Okay, what color did I make our ATP? ATP is green, okay. There's ATP and ATP. So in just the first step, see that black line, that dotted line? How many total ATP did you put into this reaction? 2. What color do I have for ADP? Blue?
Yeah.
How many ADP are coming out of it? 2. So we have 2 ATP going in. We have 2 ADP going out. Everybody got that? Okay, let's scroll down. What color do I have for NAD+? Orange. Here's NAD+. Here's NAD+. And that's it. So how many total NAD pluses? 2. What color do I have for NADH? Silver. There it is. So how many total NADHs do I have leaving? 2. All right, let's scroll down. Looks like I have 4 ADP. You ready? Here's the count. 1, 2, 3, 4. There's the 4. For ATP leaving, 1, 2, 3, 4. And then for the pyruvate, 1, 2. And then you had two waters, but those really aren't what we're worried about.